Okay.
Yes, we can see your
Hi,
screen.
everyone. Thank you for the invitation and thank you for the opportunity to present remotely. Today, I want to show you some of the results of comparative studies of mammalian immunoglobulin loci. So we all here know the way immunoglobulin loci are organized in the genome. They are superfamilies of VD and J genes. In B cells, they undergo somatic processes such as VDJ recombination, somatic hypometagenesis, and many others additional effects, and it results in a highly diverse set of antibody repertoires. And overall, we talk about this diversity as a way to fight an extremely diverse set of antigens. They are rapidly evolving, they are constantly changing, new types of pathogens emerge, so we need something as diverse. And here we can ask a question, what role germline immunoglobulin diversity play in this process? Because we have so many somatic processes that can basically generate a lot of different sequences, does it mean that the germline diversity matter? And if yes, what is the role that it plays? So, of course, modern studies, especially ones that are focused on human immunoglobulin loci and Ig genes, tell us that germline polymorphisms are extremely important. There are various associations between polymorphisms in specific immunoglobulin genes and responses to viral diseases, to predisposition to some other diseases, and even the way our baseline antibody repertoire looks, because there were multiple studies showing that germline variations can provide very different starting set of antibodies. However, if we come back to our original representation, then we still don't know how this diversity looks across different mammalian species. There are any similarities or commonalities, and if we can see them, what challenges do they pose as for us as a community? And today I wanted to tell you some of the stories that can partially answer some of these questions. So first of all, we're starting with a gene prediction tool. So how we can identify immunoglobulin loci if we have just a genome assembly. To deal with it in a standardized manner, we developed a tool called Edge Detective. It uses two types of input. First of all, it's information about known immunoglobulin genes and also information about how recombination signal sequences look. It turns out that RSSs that flank immunoglobulin genes and drive VDJ recombination are quite conserved across at least mammalian species, so we can use the existing information. And if we find matches of RSSs in the genome, then they provide us with an approximate position of immunoglobulin loci and T cell loci as well. Then we can use the database of known immunoglobulin genes and see if matches to Ig genes also coincide with matches of RSSs. And based on this, we can extract the first set of Ig gene candidates for a given species. They would form the first tier candidates, highly similar candidates to something that is already known. If we repeat this procedure using candidate genes that were just found, we can extract less conserved Ig genes and repeat this procedure until it converges. So usually it takes 3, 4, 5 iterations until we identify all Ig genes using this procedure. Of course, not all Ig genes that might be there, but all Ig genes that such an iterative method can detect. And after that, we use Ig gene candidates. Of course, we are not ready to build germline sets. That's something that you've been discussing in this meetings a lot. However, it still provides us with an approximation of what immunoglobulin genes can look like in a given species. And that's what we are using for our analysis. Our first question was to understand what is the population diversity of immunoglobulin genes and loci across different species. Unfortunately, we don't have a lot of population data for non-human and non-model organisms, but with modern sequencing technologies and genome assembly methods, in many cases, we can generate nice haplotype resolved assemblies that also cover immunoglobulin and T-cell loci. And then we can start comparing the diversity across different species. For example, we can look at bonobo. This is our close relative, so we expect it to be similar in some way. And indeed, its immunoglobulin heavy chain locus that is haplotype resolved looks somewhat similar to what we see in humans. It has some polymorphisms. They frequently concentrated in places that undergo expansions and contractions. You see this small and pretty large mosaic repeats. We can see some individual deletions, and of course, if we zoom in, we'll see a lot of changes. However, overall, that's something that we used to see. That is not a very common situation. Sometimes we might see situations that look completely different. For example, the Greenland wool immunoglobulin heavy chain haplotypes look extremely diverse. And here we don't see a strict diagonal showing us the similarity between corresponding haplotypes. Instead, we see mosaic repeats here and there, but overall we can say that the similarity was partially lost, probably due to extensive evolution or maybe even hybridization with other closely related species. That might be a case for this specific Greenland wolf, because it also showed hybridization with other wolf species. But the similarity can look differently, and even for species with highly controlled inbreeding programs, we can also see very significant diversity. For example, this is a Piedmontese cattle, also paternal haplotypes, and here you can see that they differ a lot, and they also characterized by different number of repeat copies. But unlike bonobo, where these repeats are concentrated in relatively small places, these repeats take the whole locus. For example, this block represents the whole IGHV locus. And despite all the breeding effort, the fact that this is a very well-defined cattle breed, corresponding haplotypes can be very different. So let's look at these situations more closely. And let's start with the primates, because we can say that they are simpler. If we compare bonobo with other great apes, such as gorilla and orangutan, overall we can see that, yes, there are a lot of similar blocks, and we can relatively well reconstruct long synteny blocks. We can make some conclusions about evolutionary development, but we also should keep in mind that there are places where differences emerge. For example, if you again look at this bonobo alignment plot and concentrate on this highly repetitive part, then we can see that it encodes immunoglobulin genes that look like human genes IGHV 330. So for this set of human genes, we know that they are also present in multiple copies, and if we instruct bonobo 330-like genes and compare them with human 330 genes and also with similar genes from this other species, then we'll see that these expansions emerge independently in this species rather than retain some large similarity. So the evolutionary tree of these repeat copies show us that bonobo-specific branch is located separately from, for example, human-specific branch. And there is also a branch showing one copy per haplotype that is indeed mixed with other species. And together it shows us that these places probably reflect rapid evolution of immunoglobulin loci. And it also means that finding orthologs for these genes can be incredibly tricky because they just break this model. If you see 5 copies of 330 in one locus and 4 copies of 330-like genes in, let's say, bonobo or other species, it doesn't mean that we can reconstruct pairwise correspondence between them, saying that they are descendants of the same ancestor. Well, the real development might be more complicated, and in fact, they might represent siblings or maybe even cousins. And in this case, we also need to be careful in how we treat these genes in comparative analysis. One of the possible challenge would be assigning them gene names by orthology. That what was done for a few species, and it looks like that in this case it might complicate the comparative and association analysis because it creates pretty inaccurate basis for it. So instead, we probably need to come up with something else. Maybe it's good to treat these genes together, talk about copy number variations rather than specific instances or specific gene, because that might be important. And it's also very important for the cattle. So we already looked at one example of a Piedmontese cattle, but if we compare cattle across various breeds and also look at the closely related species such as a gaur, we see that this highly repetitive structure of the immunoglobulin heavy chain loci are very common feature. So it probably means that it emerged even before all these breeds were formed and even before the gaur was splitted from the cattle. So, but on the other hand, we also see a high diversity within the breeds. So we compared haplotypes of the Piedmontese cattle and we saw that they are very different. And it looks like that this process started long time ago, but it still continues. And this actually is very challenging because we see that this rapid evolution is happening right now. And we cannot say that this is a reference cattle immunoglobulin heavy chain locus or this one, because they are so different and sometimes they can be even 2-3 times longer as compared with each other. So it creates a lot of new questions on how we are going to study and represent this immunoglobulin heavy chain loci, because there are a lot of downstream questions related to cattle health, diseases. Right now there is a bird flu going on around and it looks like that cattle are susceptible to it. So we need to understand what's going on in this loci and specifically how to deal with them in comparative studies. First of all, we can try a genomics approach and analyze repeats units in these places. So we started with a simple method that identifies highly similar copies of the same repeats. And an example of this locus aligned versus itself, we identified 5 different types of repeats. And you can see that some of them are concentrated in small portion of the locus, and some of them, such as this dark blue, are distributed all over the locus. If we extract this dark blue type of unit across different breeds, then we can see how similar they are and what are their patterns of appearance in IGH loci. Extraction of this unit across 7 breeds provided us with classification in 10 clusters, and we saw that the conservation of the cluster is high at the beginning of the locus. It's also high at the end of the locus because you see that cluster colored in different colors here have quite conserved ordering at the end. But in the middle, we see that there is the highest diversity that are concentrated. However, we can also see that there are some repeating patterns of clusters telling us that that's probably the building block of this locus, and it can be expanded or it can be contracted, but overall we might be able to reconstruct rules according to which this loci can change. And once we can do this, we can also start talking about pangenomes and maybe even representing this loci in a more succinct manner. But also, on the other hand, it creates a lot of challenges for names for cattle Ig genes because currently they are named according to one of the reference genomes that is also highly repetitive. It's characterized by its own number of copies of these repeats. And genes in this locus are named positionally, so they were assigned with names according to their position rather than clusters they belong to. Again, if we compare IgH with genes collected across various breeds and just compute their pairwise distances, do clustering, then we'll see that these names are not the best choice for studying cattle immunoglobulin heavy chain loci. For example, we can see here clusters that, for example, represent, for example, this square, it represents genes of two names, but they are extremely similar to each other, almost 99% identity, and they represent V125 and V137, and the same with this cluster, which is 114, 127. And we also have a very complicated situation going on in this lower right corner that combines 4 types of genes, but it looks like that they all represent copies of one single gene. So it might be time to come up with new naming systems, especially for species looking like this. And it's not only cattle, it's a lot of other agricultural animals. And rather than treating each gene as an individual entity with name and with its own alleles, it might be beneficial to use information about groups of highly similar genes, because otherwise it would be extremely difficult to distinguish between alleles of different copies and make association studies that are always precede disease studies in a population. So this question and many other are a subject of interest of the newly formed TRIC nomenclature review committee at IUIS. Many of my colleagues are at this meeting, so probably it would be better to talk with them in person, but I just want to say that one of our goals is to develop transparent policies to name immunoglobulin genes in such complicated cases that represent pretty new study cases as compared to what we previously knew about immunoglobulin loci. And of course, we can suggest that this diversity is even higher if you start comparing different species. So recently we looked at high quality assemblies of mammalian species, and we predicted immunoglobulin and T-cell receptor loci and genes. And simple pairwise comparison tells us that overall we don't see similarities between immunoglobulin loci from different species at all. On average, only 2-3% of each loci can be represented as alignments, and usually they represent standard classes of repeats such as lines or LTRs, so they don't represent synteny blocks. T-cell loci are more conserved. However, as you can see, it's also not a very well-established similarity, just around 10%. And one of the questions is what to do next with this. So they are very, very different. Can we still somehow represent these loci and Ig genes in a way that we can do some comparative studies so we can see some similarities? And one way to do this is to look at the structure of this Ig loci. Our first idea is to look at patterns of repeats in genomic sequences of Ig loci because some of them look very similar to the cases that we just discussed. For example, the clouded leopard immunoglobulin heavy chain locus. If we represent its similarity versus itself, then we'll see that it looks pretty similar to what we see in cattle. However, the Ig loci are very, very different. Again, less than 2%. However, we see that the clouded leopard IgH locus is formed by repeat units. And also, the cattle IgH locus is formed by very different but still repeat units. So this repetitive structure is a common feature that can help us to compare phenotypes and genotypes of immune systems. Repetitiveness is not something that is always high. If we look at other species, for example, the snowball and the hedgehog, then we'll see that only a small fraction of the Ig loci are covered by repeats. And overall, IgH locus is a place where the highest variance in repetitiveness is concentrated. IgK and IgL are much less repetitive, and T cell loci are usually not repetitive at all. So maybe something special about IgH connections with repetitiveness. And recently, our students, Marie Paspelova and Anton Zamyatin, found a very interesting correlation between repetitiveness of IgH loci and also the effective population size of corresponding species. And this correlation is negative. So in other words, if you have a very repetitive IgH locus, then it means that your effective population size is small, and you never have high repetitiveness and high effective population size at the same time. So overall, it creates this negative trend and also allowed us to formulate the hypothesis that probably the small effective population size that is always followed by inbreeding leads to transposon activation that was previously studied for various genomes, and together it leads to restructuring of Ig loci and creating this highly repetitive structure. High repetitiveness also means that all genes encoded there are very similar because they are located in copies of the same repeat. So it also means that corresponding set of genes produce less diverse set of VDG recombinations. It also allows us to formulate a hypothesis that species with highly repetitive loci might be more vulnerable to diseases as compared to species with highly diverse VDG recombinations and V genes located in non-repetitive Ig loci. Of course, this is a hypothesis. Unfortunately, we don't have a good way to test it on the same species because this process might take tens and hundreds thousands of years. However, it's also pretty worrisome because it shows us that we already see some species with this low V gene diversity, and we also can extend this observation to species who are currently going through some bottleneck, maybe because of the loss of their habitat, maybe because of some ongoing infection, maybe because of the breeding efforts, but probably what's going on right now deeply affects the ability to find pathogens in the future. So that's one of the similarities that we found across this loci, and currently we are developing methods that allow us to continue this comparison because we are also interested in specific questions on how study different species in the context of the same antigens, and with the difficulties in orthology analysis, it might be complicated. In order to deal with these challenges, we created the comparative immunogenomics group, and again, our main goal here is to develop standards for the comparative studies. I think we have a pretty cool team. We have Klaus, who has access to multiple population samples at the zoos and conservation studies. We have Anton, who specializes in genome assembly of complicated genomic regions. Of course, you all know Corey. I do the immunogenomics part of this work, and Matt specializes in evolutionary studies. And we are also very open to collaboration, so if it looks interesting to you, please feel free to contact us. We would be happy to see if we can do something together, because as you see, this area is huge and we might not even know what to expect next. But I also want to very briefly, in the remaining 7 minutes, cover two main directions that we are working right now, and they are related to some specific cases where differences in immunoglobulins loci and antibody repertoires were found, and they currently create as a basis for these comparative studies. One of them is related again to the cattle. Yes, we work on cattle a lot because it seems to be very important and tangled. In addition to all these difficulties that cattle have, they also have unusual way to diversify antibodies. And this way is fully assisted by a gene called DA2. It's a long D gene. It contributes to long CDR3s, 4-5 times longer than we see in humans. And it also has a very special genomic sequence. You can see that it's formed by codons differing from cysteine codons by a single substitution. And during somatic hypermutagenesis, antibodies derived from DA2 can accumulate mutations leading to cysteine formation at various places. These cysteines create disulfide bonds, and instead of having just a simple loop at the position of CDR3, they might have a lot of different protein shapes. And instead of changing the amino acid composition, they change the structural shape through this process that is completely the same at the level of the mechanisms. It's also somatic hypermutagenesis and VDJ recombination at the beginning. It was suggested that this mechanism is a compensatory mechanism for the loss of gene diversity. That is kind of hopeful because previously we said that if you have repetitive Ig loci and you lose gene diversity and maybe you are screwed, but cattle actually show us that there could be additional antibody diversification mechanism that can provide the species with additional diversity. Recently, with ruminant telomere to telomere consortium, we started looking at other ruminant species, and we found that this cysteine-assisted diversity is a feature of other ruminants as well. We can see it in some deers, in giraffes, and for some reason we don't see it in such ruminants as sheep, goat, okapi, and also again in some deers. So because this is such a fundamental difference, it creates a great basis for comparative studies, and right now this hypothesis is used to reveal differences between immune responses in, for example, the elk that is highly susceptible to some diseases as compared to cattle, bison, and wild-tailed deers. And another example of comparative studies is related to mechanisms commonly known as molecular mimicry. It's not specific to antibodies, but in antibody it can be explained as mimicry that makes antibody look like surface receptor in host cells, and using this trick, antibody can easily bind with a pathogen that in normal circumstances would bind with the host cell through the surface receptor. This effect is known to take place in HIV-specific responses, but it also might be important for some other cases. And our case is about plague and ferrets and other carnivore species. So currently we no longer talk about plague because we don't encounter it in our daily life. However, carnivore species that eat rodent can get sick from the plague, but it looks like that many of them have natural resistance. However, not all of them have it, and for example, one of the Native American carnivores, black-footed ferrets, completely lost this resistance, and currently its population suffer from the plague because it's transmitted by their prey. So we started comparative analysis of ferret species that are still resistant to the plague and black-footed ferrets, and we found a really cool immunoglobulin light chain V gene that has a long CDR1 mimicking CCR5 receptor. CCR5 is present on the surface of macrophages, and there are indirect evidence that it also plays a role in infecting macrophages with Y. pestis, the pathogen of the plague. If there is an antibody that can mimic CCR5, then it can trick viruses into binding it rather than binding macrophages, and thus provide a rapid defense against the pathogen. So why we are so interested in this gene? Because we saw that it has a deleterious mutation in black-footed ferrets that are susceptible to the plague. And because the black-footed ferrets went through extreme bottleneck a few years ago, the current population is based on 7 subjects, we can say that this mutation is fixed there. And probably if we find a way to restore functionality of this gene, we still can provide immunity to the plague in this endangered species. Extending this analysis shows us that CCR5 genes are quite common across carnivorous species, and they are extremely conserved in mustelids, probably because of their diets based on rodents and for some reason in bears. And overall, it provides with an interesting case study of molecular mimicry, and we're currently working on a paper describing this analysis. And I have a feeling that this is just at the beginning. If you know more about Ig loci of different species, we will start seeing more common mechanisms of Ig gene diversification, the way genetic mechanisms create them and diversify them. And we need to keep in mind that these loci are subjects of rapid evolution and that results in extreme diversity, and we might need to come up with new approaches for comparative studies, but also to represent these genes in databases. So, of course, there are a lot of talks that cover it, so I'm very happy that the community is open to these changes. And overall, I would like to thank a lot of people involved in this work. I already mentioned some of them, but it's also telomere to telomere consortia that appear to be very open to immunogenomics analysis and all the challenges that they bear. So more specifically, I want to thank the Ruminant T2T consortium that provides us with high quality data that enables this kind of studies. It's led by Tim Smith and Ben Robin at USDA. And I would like to thank all our PhD students and postdoc who is doing all the important work here. Thank you very much. I would be happy to take questions.
Yeah, Yana, thanks. That was great. I think we have time for a few questions, so I'm going to read them to you. You can hear me okay, yeah?
Yeah, I can hear you now, fine.
Okay, great.
Do you want me to stop sharing?
Yeah, I have no control over what your screen's doing, so we're at the mercy of the AV folks. Across all these species, is IgH also near the telomeric arm of a chromosome? For example, the non-human primates was this. Oh, sorry. This is, and a follow-up question is, for the non-human primates, was the source material transformed B-cell cell lines?
Okay, so for the first question, in some cases we don't see the complete chromosomal assembly, so the connection between IgH and telomere is lost. But in cases where we see it, yes, it's located very close to the telomeric ends. There are basically 10 or 100 kilobases separating it. So that might be the reason why they are so repetitive. Again, I'm not sure how exactly it works, but probably it might be there might be a connection. For the primate cell lines, there were 2 sources, fibroblasts and lymphoblastoid cell lines. Lymphoblastoid cell lines have a lot of challenges because portions of Ig loci are lost there due to VDJ recombination. We didn't analyze them because they just have limited diversity. So the analysis that I showed you is based on fibroblasts.
Okay, the next question. For repeat blocks, have you compared diversity within repeat blocks to diversity between blocks? And do some repeat blocks have more or less variation than others?
Oh gosh, where to start? Yes, we've done it, and we see that some repeat blocks are fully formed by known repeats, for example, lines. They are very diverse. They can have immunoglobulin genes located inside it, and that seems to be a way Ig genes can travel across the locus, and they can be very, very different. These tandem structures are usually formed by repeats flanked by specific types of non-repeats. Again, it might be causation, might be correlation, but overall, we see that these blocks are more conserved, probably because they have limited way to generate new copies, usually by high similarity due to non-homologous recombination. But we just started scratching the surface. I would expect that the repeat analysis would tell us about new ways of Ig gene mechanism formations that can create Ig genes with different levels of similarity.
Okay, I think we have time for one more. So given the link to effective population size and repeat number, would you expect to see increased IGH-associated repeats within the human founder populations?
Yeah, that's a good question. So it looks like that the repetitiveness association with repetitiveness is stronger in species that recently went through a bottleneck. A reason here is a scale of 10,000 years, so we no longer see a strong association if bottleneck occurred probably 500,000 years ago. So in humans, repetitiveness is not that high. So maybe they are closer to situations where bottleneck is no longer matters. And what we see currently are cases related to all the cats. They are extremely repetitive. Bears, cattle, other agricultural animals, and for some reason, marine artiodactyls, such as whales, dolphins, and hippo. So we don't have a good coverage for the whole mammalian tree, but for all the species, the bottleneck was relatively recent. For example, if we somehow have all data for maybe ancient humans close to the founder population establishment date, we might see different Ig loci. I'm not sure if it's possible because ancient DNA is pretty fragile, and we need a lot of good, nice, high quality data to reconstruct it. But yeah, if you can show it, that would be extremely cool. And it would be a confirmation of the hypothesis.
Great. Thanks, Yana, again for taking the time and calling in.
Thank you.